welcome back to another video of STEMAC, in which we're going to talk about the Meselson-Stahl experiment. So when DNA was first discovered, the question arose that how DNA is replicated, and for that 3 hypotheses were proposed: conservative, semi-conservative and dispersive. The conservative hypothesis proposed that we know that DNA is double-stranded, right? So this hypothesis proposed that both of the strand of DNA is conserved into one molecule and a new complete different double-stranded DNA molecule is synthesized. The semi-conservative hypothesis proposed that these two DNA, the double strand of the DNA molecule will separate from each other, and each of the strand will synthesize a new complementary DNA strand, and together they will form two new DNA molecules. And the dispersive hypothesis proposed that both of the DNA strands will divide into few segments, small segments, and each of the segment will synthesize a new DNA strand segment, and they will then subsequently join together to form a new DNA molecule. So the question is, which one of these hypotheses is the correct one, right? So for that, Matthew Meselson and Franklin Stahl in 1958 conducted an experiment with this bacteria, Escherichia coli. So in this experiment, 2 nitrogen isotopes were used, 15N and 14N. This is a heavy nitrogen isotope and this is a light nitrogen isotope. So what they did was, they first put some Escherichia coli colonies into this tube, okay, and they put this only this heavy nitrogen isotope in here. So only 15 nitrogen isotope was put here in this tube, and they let the bacterial colonies replicate once, okay. So normally or usually what these bacterial colonies, what they will do is they will use this nitrogen to synthesize DNA molecule, because we know that in DNA nucleotides we have nitrogen as base. So this nitrogen is needed for the synthesis of DNA. So when they used this heavy nitrogen isotope, after their growth, the tube was centrifuged, and centrifugation is a process that lets us separate components based on their weight. So when it was centrifuged, they noticed that a DNA band was formed at the bottom of the tube, because all the DNA molecules have now precipitated at the bottom of the tube because they have used this heavy nitrogen isotope. So they were observed at the bottom of the tube. This is a DNA band. Okay, so let's say this is generation 1. Okay, so this is generation 1. Now what they did is they took some colonies from here and they put these colonies into a second tube, and this time they only used light nitrogen isotope. So they used 14, only 14 nitrogen here. So this time they also let the bacterial colonies to replicate once, and after the replication they centrifuged it. This time they also noticed a single DNA band, but it was a bit higher than the first one. The band was observed a bit higher. So they came to the conclusion that these DNA molecules have, half of them have the heavy nitrogen isotope and half of them have the light nitrogen isotope. So this is basically half is light and half is heavy. So this conclusion dismisses our conservative hypothesis, because according to this hypothesis, the both of the DNA strands were supposed to be conserved into one, right? So by that logic, we are supposed to get 2 bands, one for the heavy only and one for the light only. But we got one band and it was a bit higher, which means that there are both light nitrogen and heavy nitrogen. That's why it has moved to a higher position. So this completely dismisses this hypothesis. So it's either this one or this one, semi-conservative or dispersive. So now they took some bacterial colonies from this tube to a third tube. So this was, let's say, a second generation, and this is going to be a third generation. And this time they also used the 14 nitrogen only, light nitrogen. Now after the growth and centrifugation, this time they noticed 2 DNA bands. Okay. First one was at the same position as the second one. And the second band was at a higher position, like this. So to understand how this happened, let's go back to our basic. Okay. So let's say this is a DNA molecule. Okay, this is a double-stranded DNA molecule. So when a DNA is replicated, what do we see? We see that both of the strands separate, right? So the strands will separate and they will form a complementary new strand with it. So something like this, right? This was what we saw in the second tube, where one strand is the old one and the second strand is complementary new strand. So when we saw what we saw in the second tube is that both of these heavy and light nitrogen isotopes were together in one DNA molecule. That's why they formed a one single band. However, in this third tube, when we saw two DNA bands, what happens is that, let's say, if we further replicate this, so we will get 2 for this one and 2 for this one. So as the first step, these 2 strands will also separate. So we will get something like this. Now these 4 strands will then synthesize their own new complementary strand. So we will get something like this. Now notice here, it's 50-50, right? These 2 and these 2. It's 50-50. This is what we saw, they observed in the third tube, where these 2 DNA molecules, which were the first one at the same position, they were at the same position, and these 2 were at the higher position because these 2 DNA molecules had only the light nitrogen isotopes. So they were at the higher position, and these 2 had both light and heavy nitrogen isotopes. So this is just the light nitrogen isotope, contains just the light nitrogen isotopes, and this contains both light and heavy. So after this observation, this hypothesis was also dismissed, because according to this hypothesis, the DNA segments were supposed to be in one molecule. So both the light nitrogen and the heavy nitrogen isotope were supposed to be in one molecule. They weren't supposed to separate, right? So we were supposed to get just a single band in this tube as well, but we got 2 separate bands. One for only the light one and one for the both light and heavy one, as we saw here. Now they further took some bacterial colonies from here and put them to a 4th tube. And this time they also used 14 N only, nitrogen only. So this time they saw two bands, just as this third one, but the difference was that this time this band was thinner. This band was thinner and the red band was thicker than before. But they were at the same position, okay? Just the width was a bit different than the previous tube. So what's the reason for this? Okay, let's see. Now, if we further replicate these 4 DNA molecules, what will we get? What we see here, only these 2 DNA molecules are same, and these 6 DNA molecules are same, because these 2 DNA molecules contain both these two. These 2 DNA molecules contains both the heavy and light nitrogen isotopes, and these 6 DNA molecules contains only the light nitrogen isotopes. So this is the ratio here is 75:25, right? And this is why this band here that contains both the light and heavy nitrogen isotopes was thinner, and this one that contains only the light nitrogen isotope formed a thicker band. So if we were to take the nitrogen isotope, take the bacterial colonies into another tube with 14N, we will see that this band will get thicker and this band will get thinner. So this is how they proved that semi-conservative hypothesis was the correct one. So that's all for this video. Thank you for watching.